Well, hello everyone. I'm John O'Brien. I'm in the Department of Ophthalmology and Visual Science, and as you can probably surmise from my position in ophthalmology, my research is focused on vision. If you'll change the slide to the next slide. All right. So my research is focused largely on visual adaptation, uh, processes that occur when the retina adapts to light, and in particular on the role of electrical synapses and the mechanisms of electrical synapse plasticity. So this a good example is in the top right of this slide. Many neurons in the retina many neurons throughout the central nervous system are connected by gap junctions, which form electrical synapses, and they're collected into groups. These can undergo dramatic plasticity anywhere in the central nervous system. Here we're showing a neuron in the rod pathway that undergoes more than an order of magnitude changes in its degree of coupling, and these are directly associated with the light adaptation. Those changes help the retina adapt from dark nighttime conditions to bright daytime conditions. We work out the signaling pathways that control that. In this case, these are controlled by phosphorylation of the connexin protein that forms the electrical synapse, connexin 36. The entire signaling pathway that you see on the right side of that diagram was worked out by a GSBS graduate student, Wade Kothman, who's now on faculty at American University. Among the signaling pathways worked out there is a very important role for calcium signaling and CAM kinase phosphorylation in opening those channels. And you can see below that we develop biosensors, in this case, a calcium biosensor here expressed in a transgenic mouse. You're seeing in retinal slices, and we can investigate, in this case, the role of calcium in triggering signaling at those cells. That diagram on the bottom left is showing you some information about the involvement of NMDA receptors at individual gap junctions. Each of those traces that you see is an individual gap junction responding to glutamate. We do a lot of cell biology in the lab. We've recently found a rather profound involvement of remodeling of the actin cytoskeleton in the entire signaling pathway that's shown there on the left. There's a great fantasy diagram on the right showing some current projects that we're looking at, in which we have learned that the signaling pathways that control actin cytoskeleton remodeling profoundly affect these signaling that regulates the coupling of the connexins. And so there are a number of projects going on now that are available for a student to come in and study, examining this role of actin, or of the actin cytoskeleton in signaling. Finally, in a completely different part of the lab, we also study mechanisms of regeneration of photoreceptors, in this case in zebrafish. Unlike mammals, the zebrafish has the ability to regenerate neurons that die. And so in a transgenic model of retinal degeneration that we developed, we're studying the regeneration of photoreceptors from retinal progenitor cells. So there's some single cell transcriptomic data shown there. The little cluster that's outlined with a black circle is the retinal progenitor cells, and the bits of data you see to the right are showing expression level of a few genes related to proliferation, and how they differ in the retinitis pigmentosa model. The left side of those violin plots, or the pink side, versus the wild type animals on the right side of that diagram. I forgot to mention, from the left, we have a new Brain Initiative grant to study connexins in retina to make these biosensor constructs for the 4 or so major connexins, and make tag constructs for all of the known connexins expressed in neurons. Those different connexins we believe to function in very specialized niches, very much analogously to the way different glutamate receptors function in distinct niches within different types of synapses. So that is a little summary of the types of projects that we do in the lab, and I'd be happy to take any questions.